I'm sure you've all heard this. So the randomized trial is the gold standard research design for evaluating treatment efficacy. And much of what we've been talking about this morning have been designs that are lower down on that pyramid. So I'm going to come back to this at the end and try and understand why exactly this statement is, I think, true, but there's some discussion. So what are our key questions? The first thing is, what exactly is a randomized trial, and why is it at the top of the pyramid in terms of treatment efficacy? So here's a basic scheme of a randomized trial. So we have the study sample. They undergo randomization to either an experimental group or a control group, and then we assess the outcomes. So we're going to look at each of these and consider some of the key features. So the first thing is that the study sample is a group of patients with a target disorder. And then the second point in this box is what I think is probably the more important thing, and that is that prior to enrollment in the trial, the patients need to be eligible to receive either treatment being evaluated in the trial. And if you think about it, when making inferences from the studies, one of the challenges when you're looking at comparing case series or cohort studies is that the groups may not have patients that would have been eligible for either of those treatments. But because eligibility in a randomized trial occurs prior to randomization, by definition, you have to be comfortable with the patient getting either of your arms before they're enrolled in the study. What randomization is the process of allocating subjects by chance to either intervention group. And this is a very, very key concept, and that is that of allocation concealment. And I would argue that if you don't have allocation concealment, you don't truly have a randomized trial. So what allocation concealment is a maintenance of the integrity of the randomization sequence such that subjects and/or investigators cannot know or predict which group they're going to be assigned to prior to enrollment in the study. Why is this so important?
Bias.
So bias. So what type of bias does it introduce? So let's say I'm doing a randomized trial of an open versus a laparoscopic appendectomy. And I, as a surgeon, have a fairly strong opinion that the laparoscopic appendectomy, has a shorter hospital stay. If I know my patient's most likely to be randomized to open appendectomy next, I'm not going to recommend that patient to the study. Whereas if I know that the next patient's going to be a laparoscopic appendectomy, I might be more likely to enroll the patients. So it introduces bias. How does allocation concealment differ from blinding? Okay. So what allocation concealment is keeping what that group assignment is going to be prior to the patient being randomized, prior to enrollment in the study. And it ensures that your two groups, your intervention and your control group, are determined purely by chance alone and not by some form of selection bias, which would be introduced if the allocation concealment's not known. Blinding occurs after randomization. So it's after the patient enrolls in the trial. It's keeping the group assignment either from the patient or the investigators or the people assessing the outcome. And what blinding does is blinding prevents knowledge of group assignment from impacting the outcome assessment in the study. So allocation concealment, if you don't have allocation concealment, there are many people would argue that that's not a valid randomized trial, because the fundamental thing is that patients are allocated by chance and no other factors. And it has a huge impact in terms of biasing the results. Blinding has a much smaller impact in altering the results of a trial, and is not a necessary component for a randomized trial. And in many surgical procedures, it's completely impossible to blind all the groups. For example, if you're comparing an operative versus a non-operative approach, it's going to be very, very difficult to blind the patients, although there was at least one trial in orthopedic surgery where they used sham incisions to blind the patients. So the next thing is looking at the intervention itself. So the key thing here is that the groups must only differ in terms of their care with regard to the intervention of interest. And so, for example, if you're comparing two surgical procedures, it's important that the remainder of care that the patients receive are standardized as much as possible. And that way you can make at the end of the study an inference that it's the procedure that made a difference and not anything else after the procedure that resulted in the difference in terms of outcomes. So when we look at it in terms of length of stay as one of an outcome after a randomized trial, it's very important that you have standardized definitions for when patients go home, criteria that patients need to meet in order to go home, so that it's not determined by just, clinician preference, which may then be impacted by what the procedure the clinician knows the patient had in the setting of a non-blinded randomized study. So how do we choose the interventions that we're going to study in a randomized trial? So at least in North America, we have this principle called Equipoise, and that is a difference of expert opinion as to the relative merits of treatments under evaluation. So both treatments have to have some sound evidence or at least a very good rationale for their use. So we can never do a randomized trial of two treatments for which we are certain, and there's great evidence that one treatment is better than the other, because if we know that one treatment's better, we don't really have an ethical justification to randomize patients to a treatment that might be inferior or that we know is inferior. And then the final part of the trial is after we've administered the intervention is we need to assess the outcomes. And again, standardized methods for outcome assessment. And much in the same way as in the case series talk, you saw what a difference the definition of infection postoperatively can make in terms of the overall incidence. It's very, very important to standardize your outcome assessment. So here's an example of a randomized trial from the orthopedic surgery literature. So the patient population, the subjects were patients undergoing hip arthroplasty. The intervention was randomization to prosthesis, either a polished prosthesis or a grit-blasted prosthesis, with the outcomes, and these were largely quality of life outcomes, assessed at 4 years postoperatively. So, coming back to the statement I started the talk with, being that the randomized trial is the gold standard for evaluating treatment efficacy. And the question comes is why? So I mentioned confounding before, and what confounding is that the impact of an intervention is altered or biased because of the association of the intervention with other factors that influence the outcome. Consider this study design. Patients undergoing hip arthroplasty with the prosthesis chosen by the surgeon, same two prostheses as in that trial, with the same outcomes assessed at 4 years. What are the potential confounders in this trial? Yeah, so what things might influence selection?
The surgeon may choose more appropriate candidate for one type of implant versus the other.
Mm-hmm. Okay.
They may like implant one than the other.
Mm-hmm. Someone on the side? Okay, so things like age, again, getting to the appropriateness, gender, body mass index, activity level, something as well, just surgeon skill with one particular procedure over another may have a very, very significant impact on outcome. So what randomization does is it ensures that your treatment groups at baseline are balanced with respect to both known, so those confounders that we can predict, but also unknown confounders, because your groups are determined by chance alone. That baseline equivalency of the group allows for very accurate quantification of the relative merits of the various treatments, provided that the intervention is standardized, and really the remainder of care is standardized so that the treatment only differs in terms of the intervention of interest, and that the outcomes are assessed in an unbiased manner, exactly the same for both groups. So to summarize, a randomized trial is a research design whereby subjects are assigned to the intervention groups based on random chance, and aside from the intervention of interest, care and assessment of outcomes for the various groups are standardized. So the key features in my mind is that all the subjects need to be eligible for either intervention. The assignment needs to be based on chance alone, with concealed allocation being absolutely critical, with, aside from the intervention of interest, care between the groups being standardized and outcome assessment standardized.